In an era where antibiotics have become a cornerstone of modern medicine, their unintended consequences are beginning to surface with alarming clarity. A groundbreaking investigation has revealed that even brief exposure to antibiotics can trigger long-lasting resistance within the human gut microbiome. This discovery is not just a scientific curiosity but a wake-up call for global health systems grappling with the escalating threat of antimicrobial resistance. The study, conducted through meticulous longitudinal analysis, provides a stark reminder that the tools designed to save lives may also be sowing the seeds of future medical crises. Antibiotics, once hailed as miracle drugs, are now under scrutiny for their role in reshaping the delicate balance of microbial ecosystems inside the human body. For decades, scientists have warned about the dangers of overusing these medications, pointing to the rise of drug-resistant pathogens as evidence of their misuse. Yet, much of this focus has been on the pathogens themselves, bacteria that cause infections and evolve defenses against treatment. What this new research reveals is that the story runs deeper. The human gut, home to trillions of microorganisms collectively known as the microbiome, serves as a breeding ground for resistance traits. These traits, once established, can persist long after the antibiotic treatment has ended, creating reservoirs of resistance that could one day transfer to harmful bacteria. The findings of this study emerged from a rigorous examination of the gut microbiome's response to ciprofloxacin, a commonly prescribed antibiotic. Participants, all healthy adults, were given a standard 5-day course of the drug, mimicking real-world scenarios where antibiotics are used to treat infections. Over the following months, researchers collected stool samples to track changes in the microbial composition and genetic makeup of the gut bacteria. Using advanced genomic sequencing techniques, they uncovered a startling pattern. Resistance mutations arose independently across diverse bacterial species, driven by the selective pressure of the antibiotic. These mutations, concentrated in a gene associated with fluoroquinolone resistance, proved remarkably persistent, enduring for weeks and potentially longer. What sets this study apart is its ability to capture the dynamics of resistance evolution in real time. Previous efforts to understand antimicrobial resistance often relied on laboratory experiments or animal models, which, while informative, failed to replicate the intricate interplay of microbes within the human body. By contrast, this research offers a window into the actual processes unfolding in the gut, revealing how bacteria adapt to survive in the presence of antibiotics. One of the most striking observations was the absence of fitness costs associated with resistance mutations. In evolutionary terms, this means that resistant strains of bacteria do not suffer any disadvantage compared to their non-resistant counterparts. As a result, these strains can thrive and dominate the microbial ecosystem long after the antibiotic has left the system. The implications of this discovery extend far beyond the individual level. The gut microbiome is a complex community where commensal bacteria, those that live harmlessly within the body, coexist alongside potential pathogens. When commensal bacteria acquire resistance traits, they can act as silent carriers, harboring genes that may eventually transfer to disease-causing organisms through a process known as horizontal gene transfer. This transfer mechanism allows resistance to spread across species, compounding the challenge of treating infections. The study underscores the dual nature of the gut microbiome. It is both a protector and a potential threat, capable of nurturing health while also fostering resistance. Another critical insight from the research is the predictability of resistance outcomes based on initial microbial conditions. Populations of bacteria that were abundant before antibiotic exposure and experienced significant declines during treatment were more likely to develop resistance. This correlation suggests that understanding the starting composition of the gut microbiome could help forecast the likelihood of resistance emerging. Such predictive capabilities could revolutionize how antibiotics are prescribed, enabling healthcare providers to tailor treatments to minimize resistance risks. For instance, interventions aimed at preserving specific bacterial populations might reduce the selective pressure that drives resistance evolution. The persistence of resistance mutations raises pressing questions about the long-term impact of antibiotic use on human health. While the immediate benefits of antibiotics are undeniable, their lingering effects on the microbiome could have profound consequences. A gut microbiome dominated by resistant bacteria may compromise the body's ability to respond to future infections, as these bacteria could outcompete more vulnerable strains. Moreover, the transfer of resistance genes to pathogens could render existing antibiotics ineffective, forcing the development of new drugs at a pace that struggles to keep up with evolving threats. Addressing this issue requires a multifaceted approach that goes beyond simply reducing antibiotic use. Improved stewardship programs, which emphasize the judicious use of these medications, are essential. At the same time, technological advancements offer promising avenues for mitigating resistance risks. Tools like Polypanner, developed specifically for this study, enable researchers to identify genetic changes across time, providing a clearer picture of how resistance evolves. Expanding the use of such tools could enhance our ability to monitor and manage resistance at both individual and population levels. Public awareness also plays a crucial role in combating antimicrobial resistance. Many people remain unaware of the long-term consequences of antibiotic use, viewing these drugs as harmless solutions to minor ailments. Educating the public about the hidden costs of antibiotics could encourage more responsible consumption and foster a culture of prevention. Emphasizing the importance of completing prescribed courses, avoiding unnecessary prescriptions, and exploring alternative treatments when appropriate could collectively reduce the selective pressures driving resistance. On a broader scale, the findings highlight the need for global cooperation in addressing antimicrobial resistance. Resistance knows no borders, spreading rapidly through international travel and trade. A coordinated effort involving governments, healthcare systems, and research institutions is essential to tackle this shared challenge. Investing in research, developing new antibiotics, and exploring innovative therapies such as phage therapy or probiotics could provide additional tools to combat resistance. Equally important is the need to strengthen surveillance systems, ensuring that outbreaks of resistant infections are detected and contained swiftly. One of the most intriguing aspects of the study is its potential to reshape how we think about the gut microbiome. Traditionally viewed as a passive bystander in the context of antibiotic use, the microbiome is now recognized as an active participant in the evolution of resistance. This shift in perspective opens up new possibilities for intervention. For example, strategies aimed at restoring microbial diversity after antibiotic treatment could help counteract the dominance of resistant strains. Probiotics, prebiotics, and fecal microbiota transplantation are among the approaches being explored to replenish beneficial bacteria and restore balance to the gut ecosystem. The study also sheds light on the limitations of current models for studying antimicrobial resistance. Laboratory experiments and animal studies, while valuable, often fail to capture the complexity of the human microbiome. By focusing on real-world scenarios, this research provides a more accurate representation of how resistance evolves in vivo. Future studies building on these findings could refine our understanding of resistance mechanisms and identify novel targets for intervention. For instance, investigating the role of other genes and pathways involved in resistance could uncover additional vulnerabilities that could be exploited to combat resistant bacteria. Ultimately, the study serves as a sobering reminder of the interconnectedness of human health and the microbial world. The gut microbiome, once dismissed as a mere collection of microorganisms, is now understood to play a vital role in maintaining health and resisting disease. Disrupting this delicate balance, even temporarily, can have far-reaching consequences that extend beyond the individual to affect entire populations. As the global community grapples with the growing threat of antimicrobial resistance, this research underscores the urgent need for action. By adopting a more holistic approach to antibiotic use and investing in innovative solutions, it may yet be possible to mitigate the risks posed by resistance and preserve the efficacy of these life-saving drugs for future generations. In conclusion, the revelation that short-term antibiotic use can lead to persistent resistance in the gut microbiome marks a pivotal moment in our understanding of antimicrobial resistance. It challenges us to rethink how we use antibiotics and calls for a paradigm shift in how we approach healthcare. The gut microbiome, once overlooked, is now recognized as a key player in the battle against resistance. By safeguarding this fragile ecosystem and harnessing its potential, we may find new ways to protect ourselves from the looming specter of untreatable infections. The stakes are high, but the opportunity to make a difference is greater still. This groundbreaking study highlights just how much impact even a short course of antibiotics can have on our gut health and the rise of antibiotic resistance. It's a wake-up call to rethink how we use these life-saving drugs and take steps to protect our microbiome. If you found this video informative, don't forget to give it a thumbs up and subscribe for more deep dives into the science shaping our world. Until next time.